how the summaries are given. So would you like to start your summary journey? I would have asked Wazia, but she's already suffering from bad throat. Are you there, Yusuf? Am I audible, people? I think Yusuf cannot hear me. Okay, then I will go to Devangi. Devangi, are you there? Because let's spare Wazia for today.
Yes, sir.
Yeah, thank you.
So in his experiment, T.W. Engelmann splitted the white light using a glass prism, and he obtained the 7-colored spectrum in he basically took the container having green algae spread all over and also aerobic bacteria.
Yes. Yes,
The
continue. Sorry.
bacteria mainly accumulated in the blue or the red split spectrum.
Hmm. Why? What does it tells us? What information does it give?
So he concluded that out of the white light or the sunlight, the main components of light that are required for photosynthesis are the blue and the red wavelength lights.
Yes, because wherever so these wavelength lights will have more photosynthesis done by green algae, and if there is more photosynthesis, there will be more release of oxygen, and aerobic bacteria require oxygen, so they will be present in more numbers in those pockets where there is more oxygen locally present, right?
Yes, sir.
Yeah, so that is the complete answer. So you have to write the 7 wavelengths, all 7 different wavelengths in the white light contains different energy. Okay, so he wanted to check at which wavelength photosynthesis is maximum. Now photosynthesis will be done by green algae which are spread throughout, but if it really depends on the wavelength, it will be different in different pockets where different colored light is falling. So he figured out that if it is different, if some pockets will have more photosynthesis by green algae, there will be more release of oxygen, and we can know that by looking at the concentration of aerobic bacteria in that pocket, in that region. Okay.
Yes, sir.
Yes, continue please.
Then we studied the site of photosynthesis, and basically the thylakoid membrane of the stroma lamellae and grana of the chloroplasts is the site of photosynthesis. Chloroplasts are a type of plastids which contain the green colored pigment called chlorophyll. It can be chlorophyll A or B.
Mhm. Yes, then.
And yes, sir. And photosynthesis consists of two types of biochemical enzymatic reactions, that are light reaction and dark reaction.
Mhm.
So, the membrane system, thylakoid membrane of the chloroplast, is responsible for trapping the light energy and convert it into chemical energy, that is ATP and NADPH. So this part of the photochemical reaction is known as light reaction because it is dependent on light and it can be only carried out during the day. And
Yes.
as a result of the light reaction, the ATP and NADPH is synthesized in the cell. These are
Mhm.
basically the energy molecules which contain energy in the form of bonds.
Mhm.
Then the other chemical reactions in the cell are not directly light driven, but they are dependent on the products of light reaction, ATP and NADPH. So to
Correct.
distinguish these reactions from light reaction, they were called dark reactions. But this doesn't mean that the dark reaction occurs only in darkness. They can happen during day and night both.
Yeah, so why they are called direct reactions? In just in contrast, okay, to the light reactions.
Yes, sir.
Yeah.
Then we did how many pigments are involved in photosynthesis. So we have four pigments: chlorophyll A, that is bright green or bluish green in color. Then chlorophyll B is yellowish green, and xanthophyll is yellowish, and carotenoids are yellowish orange.
Yeah. So this is the color of the pigments, right? This is not the color that they absorb mostly. This is the color that the pigments have. Okay.
Yes, sir.
Yeah. Then, in the end, we discover we studied this diagram. What does this tell us?
Yes, sir. So we had three graphs. The first graph shows the absorbance level of light by different chloroplast pigments according to their wavelengths. Uh, so we concluded that the maximum absorbance was 410 to 480 nanometers, that is the bluish green range. And then second was 650 to 680 nanometers, that is the reddish range. Means
Yeah.
the most light is absorbed in bluish green and reddish rays.
Yes. So can we say that these graphs are actually kind of helping or proving the Engelmann's experiment, the results that
Yes,
Engelmann
sir.
inferred? Yes. Right. What is second and third graph? Quickly.
Second graph shows the rate of photosynthesis. So the maximum rate of photosynthesis that is determined by the release of oxygen per time period. So it is in the 430 nanometer and 680 nanometers. And the minimum
Yes.
rate of photosynthesis of light is at 580 nanometers.
Yes.
Then the third graph basically shows the very strong positive correlation between the rate of photosynthesis and absorption of pigments, because the maximum of rate of photosynthesis is same 410 and 680 nanometers.
Correct. So this is the overall summary. Thank you so much, Devangi. Iram has also joined. Good evening, Iram. How are you?
Good evening, sir. Yeah, I'm good. How are you?
Yeah, I'm doing better, but I think I have a throat infection, so please bear with my voice. Okay, I'll not be able to be very loud today, but I hope it is loud enough. Correct. You all can
Yes.
hear me. Okay.
Yeah.
Okay. So today we'll be diving deeper into these um, chemical reactions. So I'll first start with the light reaction. So let's try to understand light reaction in detail. And this part is important for your entrances. And if you pay attention, you will just understand the scheme. So again, your memorizing stuff load will decrease. So light reaction. So as it says, so it's a photochemical reaction. The word photo means light, and the chemical part is because it's the light energy is being converted to chemical energy to make products which are ATP and NADPH. These are called high energy chemical intermediates. So in light reaction, photochemical reaction happens, as a result of which the plant cell produces ATP and NADPH. Okay. Now ATP and NADPH, these two are high energy chemical intermediates. Why are we calling it intermediates? Because they never stay like that in the cell. They are always used, they are always broken down in some or the other process so that their energy can be utilized. And it makes sense also. You never make energy so that you can store it forever. You will use that energy somewhere. So this is a chemical energy. Now, before we understand um, the process of light reaction, we have to understand what is. So there is something called a light harvesting complex. Because in light reaction, what are we going to do? What is the plant going to do? Harvest the light energy, correct? So it does not just happen on its own. There is a complex, there is a biological complex of cells, different kind of cells, with which contains pigments, and most importantly, that's the chlorophyll A and B and xanthophylls and carotenoids, pigments which are present in these cells. So in the light harvesting complex, there are something called photosystems. Okay, so um, there is photosystem 1 and there is photosystem 2. Now one thing I want you to remember is these are not named in the series of their functionality in the plant. These are named in the sequence of their discovery. Okay, so that's where it's opposite. Photosystem 2 comes first. So never get confused. I still believe that they should have changed it when they got to know it. But photosystem 2 comes first and then comes photosystem 1. Okay. So these light harvesting complexes are present in both these photosystems. So this is just, let me start the scheme here. So to begin with, we have, let me use this color. So we have, let's say, sun. Okay. And what is coming from the sun? Sunlight. Can you all see this font? Of course, can you all see this color?
Yes, sir.
So it first falls on these cells, which are called light harvesting complex cells. Now, before we go into this, let me separately tell you about light harvesting complexes. So what happens is there are cells present in this complex. So let's say these are all the cells present. So on these cells, your sunlight falls, and sunlight contain photons, right? What are photons from your physics? Photons are energy packets of light. So these photons are absorbed, and the light energy is basically coming through photons. Okay, so these cells are containing pigments. So they are pigment containing cell, chlorophyll A, B, etc. When they encounter photons, then the light energy is transferred between certain cells in a manner. So this is called reaction center, where this actual reaction is happening. So the reaction I'm going to teach you here, but know that these cells do this photochemical reaction, and then they send energy in the form of excited electrons to something known as primary acceptor. Do you understand what is happening? So overall scheme is photons will fall on certain cells. These cells will have pigments, chlorophyll A, B, the photosynthesis pigments. And then in the reaction center, this biochemical reaction will happen where light energy will be converted into chemical energy, and that energy in the form of electrons will be given to primary acceptor, which then will help in formation of ATP and NADPH. Is it clear?
Yes, sir.
So through primary acceptors, you make ATP and NADPH. Okay. Now we are trying to look at things in detail here. So let me Sorry, let me make it a little big. Let me start from here. So we have sun, from where comes the sunlight, which is the energy, and it first falls on these light harvesting complex. So these, I'm writing it at LHCs, light harvesting complex. Now this light harvesting complex, what is happening in this light harvesting complex is you are using the sunlight to split water. Sorry, you're using the sunlight to excite the electrons, and these electrons that you are exciting is coming from splitting of the water. So splitting of the water we will talk about a little late in this debate, in this discussion. So let me first focus on why I'm so bad at drawing things. I don't know, but I'll get. So this LHC, sunlight falls on this, and this LHC has some electrons in it, let's say. So there will be electrons present and they will get excited. And when electrons get excited, after getting energy, what happens? Chemistry. What happens if you give energy to electrons? People.
So electron
Yes.
jumps into the outermost orbital.
Yes, what is that process called? So from ground state, electrons goes in excited state, right? That's
Yes,
called
sir.
excitation of the electrons. If you excite it enough, it will leave the atom and will make the atom positively charged, because one electron, one negative charge is gone. So if it was neutral to begin with and you remove one electron by providing it with energy to the electron where it leaves the orbits, then it is positively charged electron. Correct? You have
Yes, sir.
studied all of this, right? So the electron gets excited and goes to an electron. So there is an electron acceptor. So it excites to a level where this electron goes to a higher state where an electron acceptor needs to catch this electron. And when this electron acceptor takes it, this electron acceptor has a high energy now, right? Because it is holding a high energy electron. Now what it will do is this high energy electron will be sent into a system called ETC or ETS, electron transport chain or system. So its short form becomes ETC or ETS. In this electron transport system, we are making ATPs. So ADP, which is adenosine diphosphate, plus one inorganic phosphate comes, and what leaves is ATP. So what is happening here is that this energy from this electron is helping to join these two molecules, and when two molecules join, a bond is formed, and a bond is nothing but a representation of energy stored between two atoms, right? When you make two atoms form a bond and make a molecule, you are storing energy between them. Yes or no?
Yes, sir.
Yes, right. So we started with the LHC. Sunlight fell on a LHC. That LHC had electrons. Now, where did this electron came from? Let me also tell you. It came from splitting of water. So remember, plant takes H2O and the oxygen it does not uses, right? So it splits this water into two electrons plus two protons, which is hydrogen plus 1 nascent oxygen. So if you balance it, it always happens with 2 molecules. So O2 is released. So this is released as O2, and this is the electron produced from splitting of water. I'll teach you about splitting of water in some time in this chapter. This is the electron which goes to this LHC, which is present in this LHC. Is it clear, everyone? If you are wondering where do this supply of electrons is coming from. So sunlight in the LHC is utilized to excite this electron which came from splitting of water. This gets excited, goes to an electron acceptor which has high energy now. So this electron acceptors, when it will give the energy into the electron transport system, when it will go through electron transport system, some energy will be used to make ATP from ADP and PI. Is it clear? So this is one thing that we made, but still it has some energy left. So that energy will go and excite another bunch of LHCs. Okay, these are another second light harvesting complex. And then again, just like normal, I'm removing this part. I need this space thoda sa. I'll draw it again. Okay, so yes. So as a result, what happens? Whenever a LHC gets energy, there will be electrons here as well, right?
Yes,
Again,
sir.
they will get excited with this energy and will go to another electron acceptor. Because all the energy was not used, only a fraction was used, and this electron acceptor again will give energy out, and this time the process that will happen is NADP positive, NADP plus comes and takes that electron, the energy, and gets reduced to become NADPH. Is it clear?
Yes,
we
sir.
also made NADPH. So we made 2 energy molecules through this whole process. Now, if you look carefully, and let me use a highlighter, very light shade if I can use, I will use this. Use a green light shade. Yes, if you see now, can I say that this looks like a Z, right? If you see, I'm just using this marker, you can say it looks like a N, but it also looks like a Z if we invert it, right? Can you see there is a Z scheme?
Yes, sir.
So first light harvesting complex accepts the electrons, goes up, then it comes down into electron transport chain. Another light harvesting complex gets excited, and then it goes to another electron acceptor, and then so in one ATP is formed, in the second one the NADPH is formed. So can you see it's a form of a Z? And also here the thing that you should note is that this thing, the first light harvesting complex and the first acceptor, is called photosystem 2. So these are photosystems. This is photosystem 2, which was discovered later but comes first in the functionality. And this electron acceptor along with this LHC is photosystem not synthesis, photosystem 1. So they are not named in the order of their functionality, they are named in the order of their discovery. So this is called the Z scheme. This whole thing is called the Z scheme.
So,
Of light reaction. Yes.
So sorry to interrupt, but this also looks like N, right?
Yes, it does. I said you in the beginning only that it also looks like a N, but so from LHC, just look at, but it's a Z if you just, so they call it a Z scheme, they just named it as a Z scheme because it makes a Z according to them. They could also have named it as an N scheme, but probably the reason why they call it as Z scheme, because Z, when you write Z, you have a directionality, right? This is how we write Z, correct? We start from one point, then we go here, then we come here, then we go here, right? People normally don't like N like this. People also can write N like this. First, second. So it does not have a directionality all the time. Now you can say that people also like Z like this. I've never seen anyone writing Z like this. Z always have a directionality. That's why probably it was better to name it as a Z scheme and not an N scheme. That's my speculation. Will it help you to remember this, Iram?
Yes.
Yeah. Makes sense, right? Like to me, I realized it that it always starts from this point here, okay, then goes to this point in this direction, then comes to this point with this to this LHC in this direction, and then goes again up to this point. So if you look at this directionality, it makes a Z, right? So this is called a Z scheme of the light reaction.
Sir,
Yes. If you have any doubt, please ask. And again,
sir,
yeah,
this
tell me.
ADP and PI were present in ETS.
Present in? No, yes. So ADP and PI are, so PI is inorganic phosphate. It comes from your diet. When you eat substances like banana that there is full of phosphorus, phosphate comes, like phosphorus comes into your system and you have a phosphate group. PO4 is phosphate group. Okay. So, and ADP is already present in all your cells. So we need energy from somewhere to join these. Okay. To join these, the energy that will be stored. So that's why when the next time you will break it, you will only break it into ADP and PI, which can again be joined back by, so it's like a fixed deposit. Whenever you will join that, you will invest some energy and store some energy between them. Does it make sense to you?
Yes,
So
sir.
they are present in the cell. In the ETS, electron transport system, the energy from the first electron acceptor in the photosystem 2, which comes first in the functionality, is used to make ATP, and the remainder energy goes to LHC, the second LHC. Now just by looking at it, tell me which photosystem do you think will have higher absorption maxima in terms of nanometers, and which photosystem will have a lower absorption maxima? So remember these LHC, okay, have these pigments. Remember I told you that the scheme was that there are light harvesting complex cells which have pigments, photon fall on them. Let me draw it here. So these are the cells of the LHC. Whether it is LHC first or second LHC does not matter. These are the pigment cells. Then photons fall on it. This energy is absorbed by these cells, and this is called the reaction center, where this reaction that we studied above happens. And these are uh, pigment containing cells. They contain chlorophyll A, let's say, and then they will excite electron which will go to an electron acceptor. Correct? This scheme is clear. So this
Yes,
LHC
sir.
has chlorophyll, this LHC also has chlorophyll, but this LHC is a part of photosystem 2, this is a part of photosystem 1. So because this process happens twice, in the first ATP is formed, in the second NADPH is formed, what do you think this LHC, that let's name it for now as LHC1 and this is LHC2. So LHC1, light harvesting complex 1, do you think it will have it will absorb at higher wavelength or smaller wavelength? Yusuf, what is your speculation? You have studied in physics the relationship between wavelength and energy, correct?
Yes, sir.
What is the relationship between wavelength and energy? Can you tell me? Wavelength, which is also denoted by lambda in physics. What is the relationship between wavelength and energy? Anyone remembers? Devangi?
E is equal to hc by lambda.
Hc by lambda. What is h and what is c?
C is the speed of light and h is the Planck's constant.
Constant. So very good. You said E is equal to hc by lambda, right? This
Yes,
is
sir.
energy. This is energy. Never thought that we'll be doing physics in biology class. Plants do physics, so that's we have to do physics because we are studying plants. Now it's plants' mistake that they can do physics and we cannot. They are more intelligent than us actually. So E is energy, and what is this lambda? I already told you this is wavelength. Correct? Now
Yes.
these two are constant, so we can just whatever is the constant will stay constant. So we can just make it one. H is Planck's constant, c is the speed of light, which is another constant. It always stays constant. So we can take it as k, let's say, any number k. But what is the relationship between E and lambda here? Lambda is wavelength. So wavelength is inversely proportional to energy. Can I say that?
Yes, sir.
Yusuf, Iram, Waziha, do you understand what just Devangi helped us with?
Yes,
This
sir.
formula. By this formula, we know that wavelength is inversely proportional to energy. So higher the wavelength, lesser the energy that light will contain. And in this last class, we figured out wavelength of light in nanometers. So red light has a wavelength around 700. So this is increasing wavelength. So if red, we go from violet to red. So what happens to wavelength? Wavelength increases, right? So lambda increases from violet to red. But what will happen to energy? Energy increases from red to violet. So from violet to red, energy decreases, right? So red light has the least energy and violet light has the most energy because it has the smallest wavelength. Make sense, everyone?
Yes, sir.
Yeah. So this light harvesting complex 1, what do you think? Let's call it what is the absorption peak of light harvesting complex 1 of the reaction center? It will be high energy or low energy?
High energy?
It will have a high energy, right? Then only if this will have a high energy, it will get excited, give some energy to the making of ATP, and still will have some energy left that it can give to LHC2 so that it can get excited, right? If this has low energy, how can it give energy to this one? So if you compare photosystem 2 with photosystem 1, photosystem 2 comes first. So photosystem 2 will have an absorption maxima at high energy. So high energy means low wavelength or high wavelength?
Low wavelength.
Low wavelength. So the absorption maxima of photosystem 2 comes at 680 nanometers. So it's called P680 system. So for LHC1, the peak, P stands for peak of the reaction center. So the absorption peak, let me also write it, absorption peak or absorption maxima comes at 680 nanometers. So it gets excited at 680 nanometers, which means more energy. So these electrons get excited at 680 nanometers, goes to this electron acceptor, gives energy for the formation of ATP, and still have some energy left. Now, the energy now will be less than what it was in LHC1, correct? So just by logic, tell me, what do you think the P of LHC2 will be? More than 680 or less than 680?
More than 680.
Perfect. So LHC gets excited at P700. This is the peak of light harvesting 2 complex. So the 20 nanometer of difference comes because that amount of energy has been given for this process. So photosystem 1 has a reaction center absorption maxima at 700 nanometers. So it's called P700. Photosystem 2 has a reaction center absorption maxima at 680 nanometers. So it's called P680. Will you remember that? Does it make sense and logic to you,
Yes, sir.
everyone? So please draw this and let me know when you are done. And you should also know that out of all the pigments that I have taught you, only chlorophyll A is in the reaction center. This reaction center that we were talking about, it contains only chlorophyll A, not B, not xanthophylls, not carotenoids. Rest everything else is present in other cells. Okay, and it's that all other pigments together form form other cells called antennae. But just know, antennae have never been asked, but just know that reaction center contains chlorophyll A. And this can also be seen here. Let's go to chlorophyll A. Which was chlorophyll A's? The green one, right? Or the blue one? Which was chlorophyll A? Blue one. So where have we figured out the maxima for chlorophyll A? One is at 410 and the other is at, can you see here for this blue? What is the peak for this blue? This blue line is for chlorophyll A. What is the peak?
680.
And what is the peak at which LHC1 gets excited?
680.
So does this tell you that in the reaction center, the only pigment present is chlorophyll A? Because if it was something else, the reaction peak would have been something else, correct?
Yes, sir.
Yes, please draw this and let me know when you're done. Pay attention to it. This is the most important part. If you understand that and the Z scheme, you're done. Now I have some questions for you, which you will encounter in some entrances. I'll ask them once you are done. So I give you 1 minute. Let me know when you are done. Noting this down. I can actually remove this part from here because this I have taught here. This is the light harvesting complex.
Sir,
Yes, Yusuf.
I have a little doubt.
Yes, please.
Can you explain how in photosystem 1 the maxima increased to P700?
Yes, sure. Now, so you understand what is this 700? This 700 means 700 nanometer of light wavelength. Correct?
Yes, sir.
So this 700 is 700 nanometer of wavelength of light. This 680 means 680 nanometers of wavelength, correct? Now, you understand that wavelength has a relationship with energy which is inversely proportional.
Yes, sir.
So if wavelength will be more, energy will be less, correct?
Yes, sir.
So to begin with, if this whole process of Z scheme is all about energy transfer. So let me explain you like this. So you have the choice to take some energy from me. Okay. Then the rule is that next you will go to Iram. You have to give some energy to Iram. Then you have to go to Devangi and give some energy to Devangi. So Iram is photosystem 2 coming first in the cycle. Devangi is photosystem 1 coming second in the cycle. And you are that electron, Yusuf, who's carrying this energy. And I am sunlight, let's say. Okay, we all are part of photosynthesis now. So if you have this job to do that, you have to go through both the photosystems. You will start with more energy or less energy.
More energy, sir.
So if you will start with more energy, you would like to get excited by a higher wavelength or a lower wavelength.
Lower wavelength?
Exactly, because if you get excited by a lower wavelength, you will have higher energy. So I have all sorts of wavelength. I am the sun. I give you a choice which wavelength you want to get excited from. You will say that I want to get excited from a lower wavelength so that I can have higher energy, so that when I go to Iram first, who's photosystem 2, I still can, after donating energy to Iram, I still can have something left to go and give it to Devangi, correct?
Yes, sir.
So that's where the LHC in the photosystem 2 gets excited by 680 nanometers of wavelength, which is a lower wavelength carrying higher energy. After going through photosystem 2, when it enters photosystem 1, it is left with lower energy. So the excitement that the electron of LHC2 will happen will happen at P700, because now the energy is low. Some is used already in making ATP. So that's where the photosystem 1, which is Devangi, will have excitement of electrons at a higher wavelength or a lower energy. Correct?
Makes sense, sir.
Make sense? Is it clear to everyone?
Yes, sir.
Okay.
So,
Now,
sorry, sir.
yes,
I
Iram.
have a
Yes,
question.
Iram. Yes, Iram.
So, as you said that chlorophyll A is the only pigment that takes part in photosynthesis. So, if we do
No,
not
not in the part of the photosynthesis, in the reaction center from where everything begins, where this excitement of electron takes place. In the LHC, there is a reaction center, and then there are peripheral cells. So only these cells through which the excitation happens are the reaction center. The reaction center will only have chlorophyll A. That's why it starts with 680, because the
okay,
maxima for chlorophyll A is 680. Other cells can have other pigments as well, so they help the process of photosynthesis. But you, to start the process in the initiation, in the reaction center you need chlorophyll A, because chlorophyll A was the first pigment to evolve for photosynthesis. Then other pigments came, xanthophyll, carotenoids, chlorophyll B, etc.
So these pigments do not take part in the process.
They do take part. They are accessory pigments. They help photosynthesis.
Yeah.
Okay. Yeah. But in the reaction center, you will have chlorophyll A. Make sense?
Yes, sir.
Okay. Now I have a few questions. Everyone is clear with this system?
Yes.
So my question is, when these electrons are moving from one system to another, starting from LHC in the system 2 and then going to the electron transport system and then going to the LHC of photosystem 1. When it's moving through all these systems, is the electron being used up or not?
Sir, can you repeat?
The electron which starts from here, this is the point from where the electron starts, right? This electron, it started from LHC in the photosystem 2. Am I correct? This is where it first encountered the sunlight, the photons, and got excited. This is where Yusuf started the journey from, let's say. Is it clear now? Where in this system this electron will get used up is my question. Where this electron's journey will stop and it will get used up.
So I think it
Yes,
would
yeah.
stop at any DPH.
Exactly, Iram. Correct. So this electron will only be used up at the final stage where the electron acceptor of photosystem 1 will give that electron to NADP plus to make it NADPH. You can clearly see that NADP plus is a positively charged molecule, which means it has more protons than electrons. So what it needs to get a neutral molecule, to become a neutral molecule, it need one electron. So you will give that one electron from that electron acceptor in photosystem 1, so that from a charged NADP positive it becomes a neutral NADPH. Is it clear, everyone? Nowhere in between this electron is being used up. It is just passing the energy from LHC1 to electron acceptor of photosystem 2, then to the electron transport system, then to LHC of photosystem 1, and then to electron acceptor of photosystem 1. Finally, it is getting used up in the process of making NADPH. Is it clear, everyone? So let me also name these LHCs accordingly. This is LHC2 coming from the photosystem 2. This is LHC1 coming from the photosystem 1. Is the question and the answer clear? Electron is not being used up as they pass through the electron transport system. But so between photosystem 2 and photosystem 1, electron is not used up anywhere. It is just passed on. Finally, it is used up in the photosystem 1 at the last step where NADPH forms. Correct?
Yes, sir.
And where does this electron come from? It keeps coming from this reaction. H2O splitting into electron and proton and oxygen. Now let's talk about this reaction, splitting of water. Because if splitting of water stops, the supply of electrons to the light harvesting complex of photosystem 2 will stop. Yes. So the question is, write down, how does photosystem 2 gets the supply of electrons continuously? How does? I'm writing PS2. Photosystem 2 gets the supply of electrons continuously. So the answer is it gets it through the reaction where water is split. So water, and where does water comes from? Roots. Plant absorbs water from the roots. Simple. So this water is split into, it starts with 2 molecules of water, which splits into 4 H plus 4 electrons plus O2, which gets released, right? This is the reaction from where this continuous supply of the electrons. So this is the electron which goes to PS2. Is it clear? And where does this happen? So this reaction, site of this reaction. So write down. The splitting of water takes place. The splitting of water takes place on the inner membrane or inner side of the thylakoid membrane. Inner side of the thylakoid membrane in photosystem 2 itself, correct? This is the site where this water splitting takes place. So it's obvious. Photosystem 2 gets the electron, and all the electrons which are lost from photosystem 1. And why are they lost? Because they are lost in producing NADPH. So all the electrons which are used up or which are lost, you can say which are removed from photosystem 1 are supplied by photosystem 2. And then photosystem 2 has to make sure that it keeps generating the electrons. So that generation happens by the splitting of water reaction, which happens in the inner side of the thylakoid membrane in photosystem 2. Okay. Now my question to you is, what is other things? So this oxygen is released readily. This is the product of photosynthesis. Okay, so oxygen is removed. What happens to the protons? So photosynthesis process creates oxygen. We know about it. It releases. It also creates electrons. So this is sorted. This is sorted. This is sorted. What happens to this thing? Sorry?
They get converted to ATP.
ATP. Oh, how will that ATP just needs, ATP does not need protons. It just need energy, right? Everything that ATP needs is present in the cell, ADP plus PI. Do you understand? It will be released, right? It will also be released somewhere. Now, as I told you, if this reactions happens in the inner side of the membrane of thylakoid. So can we all go back? Can you see the screen that I'm sharing, scrolling? Can you see this site of photosynthesis diagram?
Yes, sir.
Yeah. So where is thylakoid membrane? This is thylakoid membrane, right? Can you see here? Now, if on the inner side of the membrane, this water splitting is happening, then where are protons and oxygen released? In the lumen or on the outer side of the membrane?
Outer side?
If the splitting is happening on the inner side, how can you release it on the outer side? The oxygen and the protons will be released in the lumen, right? Don't you think so? Think about it. Think again. I'm giving you 1 minute, both of you. Oh, sorry, all three of you. Any? Okay, so Iram, where is the lumen and where is the outer side of the membrane? See, I'm confusing you. You are getting confused. If your fundamentals are clear, finally, where has the oxygen? Where does the oxygen needs to go?
So the oxygen should release out, right? So why does it goes in
The oxygen should
the
release.
lumen?
It should actually go out, right? Oxygen has to go like this. It has to ultimately leave the whole chloroplast, and finally it has to leave the whole cell and the whole leaf. It has to go into the atmosphere, right?
Yes, so why does it go to the lumen?
Exactly. That's where is lumen. I'm putting some mark. Let me use red. Tell me, is this lumen? Is this lumen or is this lumen? 1, 2, 3. Which is lumen according
The
to you?
second one. Second one.
Second one. Everyone understands which is lumen here? Iram, Devangi, Yusuf?
Yes, sir.
Very good. So because this is the membrane. So what is first one? Which is the outer side of the lumen? First or third? First, right? It's simple, no? People, are you confused? The first one is the outer side of the lumen, this one.
Yes, sir.
This one is the outer side of the lumen because it's the same as this one, right? Here. So proton has to be in the lumen. It is used inside the lumen. But oxygen has to be released outside of the cell. So oxygen is released on the outer side of the membrane, whereas the protons are released from the lumen. Is it clear? So protons stay here, but oxygen always comes out here so that it can leave the cell. If it gets trapped between the thylakoid membrane, it will never leave the chloroplast, and the lumen of the thylakoid membrane will keep becoming more and more and more and more oxidized. Okay. Is it clear?
Yes, sir. Clear.
So often they say on the inner side, so this is where oxygen is made but this is where it comes out and then it goes like this, completely out. Similarly, this is where protons are also made. It can stay in, it can also come out, but it never leaves the cell and goes out. It does, but that's not under the scope of your syllabus. So we'll not talk about that. Is it clear, everyone? So
Yes,
write down,
sir.
oxygen is released from the lumen. Oxygen is released from the lumen on the outer side of the membrane, to the outer side of the membrane, from where it can be released out of chloroplast and ultimately out of the plant cell. Okay. Is it clear, everyone?
Yes,
sir.
sir.
Very good. So now you will not be confused ever for all these questions, right? Just remember this scheme, remember where it is happening on the inner side, remember where is electron being produced and protons being produced and oxygen being produced. Remember the function of electron. Electron has to go to the photosystem 2. Proton can be stay in the lumen or in the cell. Oxygen has to be released outside of the cell. So accordingly, they will be released. Now, these two photosystems which we studied, they are present in this manner, in this arrangement, if both of them are present. But sometimes, or not sometimes, many a times, they do not work in a series. So plants can have only one photosystem. Sometimes plants contain only photosystem 1, and sometimes they only contain photosystem 2. Now the chances are, if plant only contain photosystem 1, it's possible. But do you think that plant can only contain photosystem 2? See, this is possible. Plant can only contain photosystem 1. Okay, there are many plants that contain only photosystem 1. So when only photosystem 1 is present, let's see how it will look like. So there are two type of photophosphorylation. Now what is phosphorylation? There is a word in your NCERT called phosphorylation. Phosphorylation is the process of adding phosphorus. Where are we adding phosphorus in this whole process, do you think? We are adding phosphorus to make ATP and NADP, right? NADPH. So in ATP, we are adding this phosphorus to this ADP, and in NADPH also there is this phosphorus, right? Phosphorylation is the process of adding phosphorus by giving that molecule energy. Is it clear, everyone?
Yes, sir.
Yes, sir.
So there are two possibilities in plant system. Either the plant only have photosystem 1, where the photosystem 2 is not evolved. So those are the plants which have evolved, which evolved earlier in time. Or a plant will have both the photosystems. If a plant have photosystem 2, it clearly will have photosystem 1 also, because photosystem 2 excite at 680, and by the end of this, the P700 get like LHC1 of photosystem 2 contains P700, so it will make a Z scheme, obviously. But if it only has photos, so this Z scheme that we studied is an example of non-cyclic photophosphorylation. Why it is called non-cyclic? Because a cycle is where you come back to the same where you began. We are not coming back here. We are starting from this LHC2 in photosystem 2, and we are going all the way to photosystem 1's electron acceptor, and then the electron is getting lost or utilized in making NADPH. So it's a one-way trip for the electron. Do you understand it's a one-way trip for the electron?
Yes, sir.
So it is called non-cyclic photophosphorylation. Make sense? A cyclic photophosphorylation will look like this. So let me start this topic. Cyclic and non-cyclic photophosphorylation. So you're adding phosphorus in the presence of photo, which is light energy. So what we studied above was a Z scheme, which was a non-cyclic photophosphorylation. Now in a cyclic, what happens is it happens under a condition when only PS1, photosystem 1, is functional or is present. Then it's called cyclic photophosphorylation, because in this case you will not make NADPH. Correct? Do you understand?
Yes.
People, how we will not make NADPH? Let's see. Again, this is sunlight. This is me. Let's say sunlight is falling on what? Only, tell me, only PS1 is there. So there will be light harvesting complex. I'm sorry. And in this light harvesting complex, if it is just photosystem 1 present, what will happen? It will have chlorophyll A in the center. And what will the P value? What will be the peak? It is photosystem 1.
700.
700.
Perfect. So I'm just mentioning it at the top. This plant just have photo system 1. So it will be P700. So it will not get excited at 680, right? Because it has just P700. Okay. 1 second. So I'll make it like this. So this LHC will get excited at P700 because it's the photosystem 1. The electron here will go up, get excited, and will reach what? Where it will reach? Electron acceptor, right? Of this system. And what will this electron acceptor do? It will take it to electron transport system, correct? Or I am making it this side, it's congested. Let's make it this side. This will take it to electron transport system. And now whatever energy you have, you will give it to electron transport system, where what happens? ADP plus Pi comes, and what leaves? ATP, correct? So we have formed ATP on these two systems. And what will happen to this? What will happen to the electron? Is the electron getting used in making ATP? Tell me. Was the electron getting used up here in making ATP? No, right? It was still passing on. So here also the electron will not use up because it does not pass on to NADP, but it comes back to the photosystem 1 ka LHC. It comes back to the light harvesting complex of photosystem 1. So the electron starts its journey and comes back to LHC of photosystem 1. So do you think there is a cyclic flow of electron here? Electron is never utilized. Do you understand, people?
Yes, sir.
That's why it's called a cyclic photophosphorylation, because in the presence of photo, this is photo, we are making ATP, which is called phosphorylation. So photophosphorylation, and the electron is cyclic, so it's called cyclic photophosphorylation, and it only happens when only PS1 is functional in a plant. But if PS1 and 2 both are there, it is a non-cyclic photophosphorylation, because you make ATP by doing phosphorylation in the process, in the presence of photo, but it never comes back. The electron is lost. So it's non-cyclic photophosphorylation. Okay. Now quickly, just tell me, what do you think is happening here? What do you think is the benefit of cyclic phosphorylation or non-cyclic photophosphorylation? What is one difference that you can clearly see between the two? One difference which I can see is that cyclic photophosphor. Yes, Iram, you tell.
So in non-cyclic photophosphorylation, there is breakdown of water, whereas here it is not.
Perfect. We constantly need water to keep breaking it into electrons and keep feeding it into PS2 so that it can keep feeding it into PS1 so that PS1 can keep feeding it into NADPH production. This is one difference, right? Versus in a cyclic photophosphorylation, we do not need constant supply because there is no NADPH formation, only ATP is formed and the electron is coming back. Yes or no? This is the first difference. So which system, cyclic or non-cyclic, needs more water?
Non-cyclic.
Cyclic. How can non-cyclic need, sorry, what did I ask? Need more water. Yes. Non-cyclic needs more water. Sorry, my mistake. So if I ask you, if I go to a desert and I look at plants, what kind of plants will I find more? The plants doing cyclic photophosphorylation or non-cyclic photophosphorylation?
Cyclic.
So these plants will have both the photosystems or will have only one photosystem?
One.
And which one will that be? Photosystem 1. So all plants have photosystem 1, whether they are living in water rich conditions or water deficient conditions, whether they are doing non-photosphorylation, non-cyclic photophosphorylation or cyclic photophosphorylation. But all plants do not have photosystem 2. Okay. And plants which only have photosystem 1 can do better when they do not have a constant supply of water, because they can keep channelizing their electron again and again and again and keep making ATPs. Does it make sense, everyone?
So I have a
Cyclic.
question.
Yes, please.
So sir, if a plant has photosystem 2 and photosystem 1 and undergoes non-cyclic photophosphorylation, so will it again go through cyclic phosphorylation or no?
Now, if it has both photosystem 2 and 1, the photosystem 2 will get activated at 680 and it will have a non-cyclic photophosphorylation. So some plants adapt later on. Because the other difference which I was talking about here, one you mentioned, the water splitting, other is cyclic photophosphorylation occurs. You can make a system having both PS1 and PS2 undergo a cyclic photophosphorylation when only the wavelength beyond 680 is available, right? So let's say I take a normal plant, but I'm only giving it red light. I'm only giving it red light at P700. Then will it undergo a cyclic photophosphorylation or non-cyclic photophosphorylation? Is my question clear?
So could you repeat, please?
If I take a plant having both the photosystems, but the only light I am giving to the plant is red light having 700 nanometers of wavelength, which photophosphorylation will take place in the plant, cyclic or non-cyclic?
Cyclic?
Why cyclic? Why not non-cyclic? It has both the photosystems.
But you're providing only 700 nanometers
Yes, so
wavelength.
only photosystem 1 will get activated and not photosystem 2. Correct?
Yes. And so
So
one more
cyclic,
question.
yes.
So in cyclic photophosphorylation, water is not broken. So in that case, oxygen will not be produced.
Yes, right. In this case, these plants will not produce that oxygen in the atmosphere. Correct? Very good. Very good point here. So it makes sense to all of you?
Yes, sir.
Very good. Okay, so we will be there are some things. So to Iram's question, let's for now take that as a very good observation, and we are now understanding that plants going cyclic for photophosphorylation do not produce oxygen, or to generalize, produce very less oxygen as compared to non-cyclic photophosphorylation plants. Now, what are the conditions where splitting of water can still take place? I'll tell you for your entrance perspective in the next class. Okay, so we will go now from here onwards to the details of why are these ATPs and NADPH produced and where they are used. Okay. So if you have any doubt, I'll stop now for questions. Otherwise, I declare the class. Please go back and revise, people. This was some technical heavy class, some mechanism heavy class, but if you revise it once, I'm sure that you will not forget it. Any questions? No. Okay. So I'll see you all in the next class. Bye-bye.
Thank you,
In the
sir.
next class, we'll be
Thank you,
doing
sir.
Kelvin cycle. Okay. So the Kelvin cycle is another cycle that you have to remember. So please go back and read Kelvin cycle from your NCERT. It is asked. Okay. It will always be asked. Kelvin cycle, which includes C3, C4 pathways.